Bismillahirrahmanirrahim. First of all, I want to introduce myself. I am Dr. Rais Ahmed, Assistant Professor, Environmental Microbiology. My topic of discussion is the journey of immunogenesis from vaccine administration to long-term immunity. We know vaccine is a basic tool that can prevent the animals or a host or human body from any type of disease. In this assignment, we will focus on how vaccine works. These diagram show that vaccine is going on in animals, in dog, cat. This is the basic of vaccination. For a good vaccine program, we must focus on age of first vaccination, type of vaccine, route of vaccination, and number of vaccines. It means how much booster doses are required for proper immunization. Then basics of vaccine program. We must know about if we want to get good immune response, then we have to choose the correct vaccine. We have to choose the correct dose, then a good amount of immunogen, then we will get a good immune response and CMA response. Otherwise, vaccine failure, vaccine will not work. This is the schedule of vaccine in cattle and buffalo in Pakistan and in this region of subcontinent. Mostly the vaccines of FMD, BF, HS, Crohn's disease, IBR, BBD, parainfluenza, anthrax, bovine rotavirus, bovine coronavirus, Brucella abortus. These type of vaccine are being administered to the cattle and buffalo. This is the vaccine schedule of sheep and goat in Pakistan and in this region. The vaccines of against FMD, tetroxemia, CCPP, PPR, sheeppox, goatpox are administered to sheep and goat. Now we discuss about the vaccine failure. The vaccines are going on in field. So we must know why vaccine is not working, why there is outbreak of disease. So there are some biological factors like force genetics, age, health status of the animal. Then there are some issues with vaccine-related factors like composition of vaccine is not good, vaccine storage and handling is not maintained, cold chain is not maintained, environment is not favorable. Maybe there is more exposure to the pathogen, and sometimes there is pathogenic evolution. There is antigenic mutation, a virus or any causative agent changes its shape. So this is the antigen processing and its impact on immune response. If we are going to design any vaccine, we must know that the antigen processing and presentation pathways. We know that if we inject any type of antigen, then it must be processed through APCs and it must be presented through APCs to the T cells, and we must activate T cells for proper immune response. The objective of this PPT to elucidate the biological mechanism by which vaccines stimulate the immune system, to investigate the immediate immune response following vaccine administration, to explore how immunological memory is established post-vaccination. This is a sketch diagram where we can see vaccine is going to inject to the body of host, then vaccine antigen is being carried by APCs. Here is the genetic cell. It is going to present the antigen. Processing of antigen is going inside the cytoplasm, and antigen is presented by class 2 MHC molecules, and it is rejected by TCR of the T cell. Then T cell plasma proliferation occur, and B cell they convert into plasma. Plasma produce a lot of amount of antibodies. That is the humoral component of the vaccine response. Then along with this T cell also get activated and they produce CMI. After injecting antigen, how is it processed inside the body of host to produce effective immunity? Let's understand in next slides. This is a simple, very simple outlet or the diagram that shows activation of TLRs by PAMs. So there are many PAMs over bacterial, viral, any type of antigen, and there is a discussion between TLRs and PAMs, and then the antigen is declared as non-self. Non-self antigen is actual antigen is non-self. So there is a discrimination between self and non-self antigen by immune cells. These are the dam-associated molecular patterns, which are also detected by the TLRs. This is TCR CD3 complex. TCR of the T cell consists of the two chains alpha and beta. They are linked with the CD3 complex. They consist of five chains alpha, five chains that contain epsilon, gamma, sigma, delta, zeta, zeta, or zeta, eta. Zeta or zeta, eta. They are linked with the items, and the items trigger signal transduction molecules. And this is how clonal energy develop in TCR. This is the immune response where MHC gene A is detected, and then if MHC-B gene is rejected, that ensure that challenge MHC compatibility issues, then we will not get any response. This is how T cells, T helper cells, and TC cells they identify the antigens. T helpers identify the antigen by class 2 MHC, and T C cell detects the antigen by class 1 MHC. This is the crosstalk between APC and T cell, and there is a compatibility of MHC is ensured, and co-stimulating molecules they crosstalk each other when CD40 of APC binds with CD154 of T cell. Then there is second crosstalk between the CD28 receptors of T cell with CD886 receptor of APC. Then there is activation. Otherwise, there is inactivation. This biochemical basis of TCR signal transduction. The outcome of the TCR, or you can say TLR PAMS outcome is the triggering of ZAP70 protein that ultimately activate NF8, NF-kappa B, AP1 signal transduction pathways, which are going to do gene transcription, and then there is production of cytokines. This is a crosstalk between T cell and B cell. B cell identifies the antigen in the free form and present antigen through class 2 MHC, and then it is detected by T cell. This is turning off the B cell due to cross-linking of parent antibodies. If the parent antibodies are present, then immune system will not work. This is the differentiation in the time course of T-dependent and T-independent response. You can see there is in T-independent immune response, there is a production of IgM only, no production of IgG. So if we want to get a good CMI immune response, we have to generate CMI response. This is how antigen is going to process by APCs. So first of all, antigen is going to be degraded into peptides by a system of proteolytic enzymes. Then it is going to be presented to MHC molecules. Then MHC molecules from the lumen of RER, they are going to be fitted with the peptide fragment, and then it is presented over the surface. This is the presentation path. We have class 1, class 2 molecules. Mostly in vaccine antigen, we focus on class 2. And this is how invariant gene of class 2 MHC molecules is going to be degraded. And then how this peptide fragment of attached to the cleft of MHC molecules, and how it is presented over surface of APC. This NF-kappa B signaling pathway. A lot of signals they are triggered. All these are cascade reactions, but we are mostly concerned with the T cell differentiation and activation. Once T cell get activated, you get a good immune response. This is the picture of the vaccine. It looks like very simple, but it's very complicated inside. There is there must be activation of B cell and T cell for good immune response. So this is my project, development of vaccine against lymphoid disease for ruminants. I am working on it as a principal investigator. We collected the sample from the field area and we are going to formulate a vaccine against KZ lymphadenitis, that is also called the lymphoid disease of sheep and goats, that gives a very huge economic losses to the poor livestock farmers. And due to my research background, I got a certificate of appreciation of outstanding highest benefactor award of research and publication in microbiology department from the University Vice Chancellor. My future perspective of this assignment, emerging areas of research, by having the understanding of all these mechanisms, we can formulate a rational vaccine design, personalized vaccine, nanoparticle based vaccines, immune modulator strategies, infectious disease research program, computational and structural biology, studying immune evasion mechanisms, biomarker discovery, and improving existing vaccines. These are the references for the assignment. Thank you for your attention.